All right, so we're moving on to Chapter 8, which is Carbohydrate Metabolism. Okay, so carbohydrate metabolism is exactly what it says on the tin. It is the metabolic processes that surround carbohydrates. Um, we will not go into the detail that is discussed in Chapter 9 right after this. Instead, we're going after we're done with this chapter, we'll move on to nucleic acids. That way, we can talk about that class of biomolecules before we move back into a more in-depth discussion of metabolism. So, the first thing you might be asking is, what is metabolism? Well, metabolism is defined as all the reactions that are enzyme mediated that sustain life. Okay, so every reaction in a cell that goes to sustaining the life processes of the cell is part of the metabolic pathway. Okay, we have two classes of metabolism. First is catabolic. Okay, and catabolic reactions are those involved in energy production. All right. The second class is anabolic. You may have heard the term anabolic steroids before, and those are used for bulking up because anabolic metabolism is biomolecule synthesis. So muscle cells, um, would be in that class. Okay. Together, together, these reactions achieve what is called a homeostasis. All right. And this is the self-regulation of internal systems. Okay. We are going to start off by focusing on glycolysis. Okay. So glycolysis takes place in almost every living cell. It's probably one of the oldest pathways in biochemistry, and both prokaryotic and eukaryotic cells have exhibited high conservation in the number of steps involved and the enzymes involved in the glycolysis pathway. Okay, so the glycolysis pathway is the one involved in pretty much the production of energy from carbohydrates. Okay, and there are two pathways available for glycolysis. There's anaerobic and aerobic. Okay, anaerobic does not require oxygen, so anaerobic glycolysis was very important on the primitive Earth because there was low partial pressure of oxygen available to the monocellular organisms. whereas aerobic requires O2. And that's what happens when you forget to put your cell phone on silent. Okay. All right. Both of these pathways, all right, start by splitting glucose. Okay, we have glucose, and it will be split into 2 moles of pyruvate. Okay. From there, the treatment of the pyruvate molecules differs based on the organism, based on whether it's aerobic or anaerobic, and so forth. Okay. Um, I think it would be a useful exercise for you to take a few minutes and calculate the change in free energy for this process to split glucose into 2 pyruvate molecules. Take a few minutes, pause the video, and do that. Okay. So, in these reactions, we get about 5% of the energy stored. Okay. And we do this in 2 moles of ATP and 2 moles of NADH. Okay. Initially, we will invest 2 ATP. So overall, we take 2 ATP, we put it into the process, we get 4 ATP out, which gives us a net of 2 ATP. Okay. So just bear that in mind. All right. So in anaerobic organisms, pyruvate is broken down into things such as ethanol or lactic acid or acetic acid. Okay. We call these processes fermentation. All right. And remember, they are anaerobic. In aerobic metabolism, pyruvate is completely broken down into carbon dioxide and water. So glycolysis occurs in 10 reactions, okay, and 2 stages. First stage is phosphorylation of glucose. Okay, we do that twice. And cleavage. And that will yield 2 glyceraldehyde, 3 phosphates. Okay, and it consumes 2 moles of ATP. The second stage of glycolysis is where we convert this glyceraldehyde 3-phosphate, which I'm just going to abbreviate as GAP. Okay? So we convert that to pyruvate. And this will yield us 4 ATP and our 2 NADH. Okay? So if we want to summarize this entire reaction, we can do it as follows. 1 mole of glucose plus 2 ATP plus 2 inorganic phosphate plus 2 NAD+, which is the oxidized form of that molecule, okay, is converted to 2 pyruvate plus 4 ATP plus 2 NADH plus 2H plus 2 H2O. So we can go over some of the reactions in detail now. And I think for this video, I want to aim to try and get through stage 1, but we'll see how far we get. Okay, so our first reaction involves the synthesis of glucose 6-phosphate. Okay, and so what we have is we have our glucose. Okay, and I'm going to just stick to drawing the Haworth projections here. Okay, and if this is carbon 1, 2, 3, 4, 5, and carbon 6 is here. Okay, that will be combined with 1 ATP molecule, okay, by the enzyme hexokinase. Okay. Kinase adds inorganic phosphate to molecules. Okay, and hexokinase requires magnesium ions to function. Okay, what we get out of this reaction is glucose 6-phosphate. And forgive my sloppy structures on the whiteboard. Okay, plus 1 mole of ADP. All right, so this would be glucose 6-phosphate. I tend to abbreviate this in my notes as G6P because lazy. Okay, so in this form here, the glucose 6-phosphate can be passed to another enzyme and used in a further reaction. Okay, So I'm just going to redraw this. Oh, I'm not drawing glucose. Okay, so this molecule is then converted using an enzyme, okay, to fructose 6-phosphate. So that is our second reaction step. Okay, and this is done by the enzyme phosphoglucose isomerase. Okay, and so what we get out of this is fructose 6-phosphate or 6-phosphate. It's still 6-phosphate. Okay, so what you can see is the enzyme breaks the ring here at this bond and it reforms it here. Okay, and that makes a hydride transfer take place, which gives us this CH2OH group. Okay, so this is fructose 6-phosphate. Okay, which I tend to abbreviate as F6. Third reaction, okay, is we need to phosphorylate fructose 6-phosphate to fructose 1,6-bisphosphate. Which I tend to abbreviate as F16B. Okay, so we take our fructose 6-phosphate and if you are writing all these reactions down in your notes, you will very quickly come to remember all the structures for those monosaccharides that I wanted you to learn. Okay, we combine this with another mole of ATP and the enzyme, okay, called phosphofructokinase 1, okay, which also requires magnesium ions And this is abbreviated as PFK1. All right. What we get out of this, and I'm going to start using the shorthand here for our phosphate group, is we get our bisphosphated fructose. Okay. But we're still not quite done. All right. We're still not quite done. Because the next step takes this bisphosphate and preps it to actually go through the pyruvate making functions. Okay. And so we have to cleave this new molecule. Okay. And you'll notice I didn't erase it. And that's because this is an equilibrium reaction. Okay. And the enzyme responsible is aldolase. That's messy. Let me rewrite that. Okay. And so the aldolase enzyme will convert this fructose 1,6 biphosphate or bisphosphate into two different molecules. Okay. The first will be the molecule dihydroxyacetone phosphate. Okay. All right. And then the other will be 1 mole of glyceraldehyde. One moment. Thousand pardons, I messed up that first structure. So dihydroxyacetone phosphate. See if Dr. Conning can actually draw this properly this time. Got a carbon. And then the other is glyceraldehyde, 3 phosphate. So this is our glyceraldehyde. Three phosphate. This is our dihydroxy. Acetone phosphate. Okay. So that is the end of stage 1. Right? The first thing that has to happen in stage 2 is we need to convert this to glyceraldehyde 3 phosphate. Right? We'll discuss that and the rest of the stage 2 reactions in the next episode, the next video, and then we'll move on to discussing the metabolism processes in a little more detail. Okay. After that, we'll talk about the enzymes that are involved a little bit in some more detail, how glycolysis is regulated in detail, and then we will go into glucogenesis. Okay? So I hope that you all are staying safe and staying well, and I'll talk to you in the next video.